Professor DeAngelis, it's a real thrill to have you back at this particular conference. You presented some extraordinary research this morning. Could you encapsulate and tell us a little bit about it and its significance?
Okay. Thank you for your kind words, and thank you for attending the talk. The real foundation for what we presented this morning really has to do with our biobank, and really has to do with the people of Utah, the organ donors, and the Lion's Eye Bank of the Moran Eye Center, but really has to do with the people and their loved ones willing to donate their precious eyes. So that enables us to do our research and further our work into age-related macular degeneration, because nothing comes close to replicating what goes on in a human eye and to recapitulating the macula. And no animal model really does that. So we can get human eyes and we get them fresh. And that's really important to really, again, use this word recapitulate to what's going on in the disease processes so we can get to disease mechanism. The work I presented this morning takes advantage of our genetic data and our expression data. So how and to what levels our genes are expressed. So we mesh that together and trying to get information at what's going on between eyes with disease and eyes without disease from different people of the community. So whether or not you have disease, it's important to donate your organs to science. And we do this and we study this. And it's important to be available no matter what time of the night people decease to accept this precious tissue. And we not only get eye tissue from people that decease, but we take blood, serum, and plasma so we can study all aspects of what's going on in the body systemically and localized. And what we're studying right now is the link between risk factors in the environment and your genome. And that's called epigenetics. So these are things that can happen to your DNA, but they necessarily don't change the nucleotide bases. And that's what we're trying to measure. And the data I presented this morning between gene expression and your genomic DNA, our preliminary data suggests there is a lot going on there. And we want to get to those mechanisms because right now in the eye, that's very understudied, whereas that's been quite studied well in cardiovascular disease, obesity, and other diseases of a complex nature. And what I mean by complex, environment and genes play a role. Type 2 diabetes is another example. So we want to bridge this link in eye diseases. And a major eye disease is age-related macular degeneration. So that's our next step. So the work I presented this morning, we'd like to get published as quickly as possible, and we're in the process of validating that right now.
So what are your next steps in that validation?
Well, we're validating as we're speaking. So it's to take it to a micro level, because we've done it at a large aggregate level, and now take it to a very tiny level at the lab bench, do it in individual pieces of eye tissue, and say what we see at the large level, can we validate it at a smaller level and in other patient samples, not just the ones we got the positive results in. And not only in our samples from Utah, but can we replicate it in samples, say, from Alabama or samples from another state? Because any therapeutic we want to identify, does it have global or national applicability. That's going to make a therapeutic successful and in other populations.
What are the key requirements to being able to look at epigenetics?
The key requirements to looking at epigenetics, number one, is to have a really good biobank that's well phenotyped, and is to have a good team of both clinicians and scientists to do that. So you have to have good tissue collection in place, fresh tissue. You can't have old tissue that's been sitting around for a while. So you have to have a good team between an eye bank, such as the Utah Alliance Eye Bank that works with the Moran Eye Center, and a good team in your laboratory to take that tissue in a timely fashion and accession it, collect it and accession it. And then we have a team of clinicians, which includes Dr. Al Vitelli, Dr. Leah Owen, Dr. Akbar Shakur, and Dr. Ivana Kim here at Mass Eye and Ear Infirmary. So we have a team with diverse clinical expertise that can diagnose these eyes post-mortem. Because while people do have disease and we may have clinical records on file, we still want to diagnose the tissue post-mortem, because there's things that can be seen in the tissue that were not seen while the patient was alive. Secondly, let's face it, most people don't go to see a doctor until they have a problem. And many people from the community are normal, or think they're normal. And they may go around with some form of disease, but they'll never go blind from that disease. And they have the early forms of the disease. Those are people we want to see, and we can learn a lot from those people. And we can learn a lot from people that have no disease whatsoever. Why are those people normal at a very old age? We want to know what's protecting them.
That's a very, very powerful way of looking at a complex problem. So tell us about some of the other work and the next steps if you have.
So we're using this very powerful repository to take this next steps to do a genome-wide level methylation to look in an agnostic or to validate our results from genomic and gene expression, to say, now we found some tissue, we found some genes and pathways that are suggestive of methylation. Can we show those same genes and pathways actually have methylation? So that is our next step, and that is what we hope to do. And furthermore, we've shown at the retina level, there's two types of tissue in the back of the eye that are important, the retina and the retina pigment epithelium. And it's the macula of each of those tissues, which are composed of, that are important. Which cells in the retina pigment epithelium has one cell type that's important. But in the retina, there's at least 70 cell types in the human retina. We've got to tease that apart. What's the best way to do that? And how do you study those cell types? We can't study the retina as a whole. We've got to tease apart. And then study those cell types for which ones are contributing to methylation. So we're going to do that first by gene expression. That will dictate which ones are contributing to the AMD pathology, and then look at methylation. Because why do some of these patients have these risk variants? They smoke 140 pack years. I showed that example of those twins.
Right.
140 pack years.
Yeah.
Every disease risk variant know in their genome, and they have no disease. What is protecting them? But his identical twin has the most severe form of the disease. He's completely blind with age the most severe form of age-related macular degeneration. What's going on? Maybe it's methylation. So we need to find what's protecting, what's causing. And methylation has actually gone to clinical trial for certain diseases, but is very understudied in eye diseases altogether.
I think it's a very powerful and important approach.
Thank you. We think so too. And that's what our hypothesis is.
That ultimately, do you see this from a therapeutic perspective? You'll have drugs that will interfere with methylation. There will be potentially gene modification therapies that would target this particular issue?
We hope so, some focused gene therapies, because there's some that are in clinical trials for other diseases, for cancers, whether it's suppressing methylation or increasing it. I don't know. I haven't thought that far ahead. We just want to identify, and the best way to identify that. Because I think that is still open, and we're going to have to, again, validate that. So, again, it comes back to the systems biology approach, using several different approaches: gene expression, genome-wide association, gene and methylation, and bring all that data together so you have complementary approaches that validate each other, and then replicate that so that you have confidence in your data.
So essentially you're creating the general theory of methylation in the eye.
Yes.
With all of the different components that might be contributing to this process.
To try to get to therapeutics for age-related macular degeneration. Then we have to deliver it. So I think there's still debate. Is this a localized disease? Is this a systemic disease? Are some aspects of the disease localized as opposed to systemic? I don't know.
This is an absolutely critical area of research, and we thank you for pursuing it so vigorously and so with such a degree of persistence and perspicacity.
Thank you for allowing me to speak to it, and thank you for allowing me to talk about my collaborators. And I also want to mention Lindsay Ferrer too, for helping a lot with the statistics and the bioinformatics of it all, because you have to have robust statistical analysis in all of this.
Professor, thank you so much, and we continue to look forward to more good work.
Thank you. Thank you for allowing me this opportunity.